Welcome, everybody, to another episode of the Beyond Train podcast. I'm your host, Liam Dalton. Today, we have a very special guest on. I'm very much looking forward to this discussion. We have Jeremy Hammond on, and we're going to talk about viruses today. I'm really looking forward to this discussion. It's been a long time coming. To be quite honest, it's been kind of difficult to get somebody on to talk about this from the opposite perspective. So I'm really grateful for Jeremy and his time today. And if anything, I think in a true pursuit of truth and in real science, it's best to understand both sides of the argument. It doesn't matter where you fall in your opinion. It's best to know both sides to be able to not only strengthen your argument, but gives you a true holistic understanding of what's going on. And so I think that's really important. So uh, if everyone keep open mind today and be receptive to this, I think that's one of the most beneficial um, things that we can do moving forward. I don't care if you're no virus or not. I think that's really important. So, anyways, I've talked enough. Jeremy, thank you so much for coming on today.
Sure. Thanks for having me.
Definitely. So the question I ask all my guests, the introductory question is, what is health? And sort of get a definition of what health is, what it means to you, how does it manifest? And this gives us a good little baseline to work off of.
Gosh, what is health? There's so many ways you could describe it and define it. I guess basically I would say that health, a state of health, is a state in which your body is functioning as it was designed to do. I guess that would be my most simple definition.
Nice and concise. Perfect.
Yeah.
That's awesome. Yeah. And I mean a great definition too, right? Because that's sort of the goal. Like here we talk a lot about symptoms and you know we shouldn't necessarily demonize symptoms, right? Because their response, their body's response, and if you're getting symptoms at the right time in as the correct response, that's sort of a good thing, right? So
Well,
um
sure. And the
anyway.
caveat to my little simplistic definition would be, yeah, it was sometimes your body functioning as it was designed to do can be harmful.
Mhm.
Immune overreactions are an example of that. So, my definition is admittedly simplistic, but I was thinking in terms of, we have this paradigm of like mass vaccination and then pharmaceutical interventions. And it's like the idea of vaccination, for example, is that it's almost as though it's premised on this assumption that we all require these pharmaceutical interventions for our immune systems to function. And this is kind of what I was getting at with my simplistic definition, was kind of just to correct that misperception.
Mhm. Yeah, sort of like the idea, and correct me if I'm wrong here, but the idea that, we are designed perfectly in a way, right? Would you agree with that?
Yeah, and I think if we give our immune systems, for example, what they need to function, that's you can maintain that state of health, and you can be challenged by pathogens and toxins, and your body can function. Your body can detox. It has an your immune system can function, and you can maintain a state of health. And the problem that I see with the population today and the epidemic proportions of chronic illnesses and so on is that people's people's bodies are not functioning as they were designed to do. There's just too much environmental uh, exposures that are just ruining people's health. And so, the body has limits in terms of what it's capable of doing, in terms of detoxification, in terms of the immune system and what it can do. And so, I guess the further caveat to my simplistic definition is we need to give the body what it needs to be able to do that function.
Mhm. Yeah, like proper nourishment is sort of what you're
Sure.
getting
Yeah.
at there.
Nutrition, avoidance of toxins, yeah, and so on. Sure. Exactly.
Okay. Just out of my curiosity, would you attribute some sort of psychological nutrition in a way, like what you consume on a psychological level? Do you think that has an effect?
Um, in terms of, explain further.
Sure, sure. So say somebody, like an example would be if somebody undergone some sort of severe trauma. If they experienced a traumatic upbringing, maybe physical abuse, and
Sure.
maybe that is a
Yeah, yeah.
physical infraction, but
Oh, absolutely.
stuff
Yeah, I
on that
mean,
level.
mental health absolutely ties directly into physical health. Absolutely. There's
Mhm.
no question. Yeah, I think so.
Awesome. Great. Appreciate that. Uh, let's dive in to today's topic, viruses. I'd love to hear. I think an important place to begin, and I'd love your input as well. If you think that there's a better starting point, maybe we could begin with virus isolation. Love to hear your thoughts on that.
Virus, I'm sorry, what?
Isolation.
Isolation. Well, sure. Yeah, there's this claim that viruses have never been isolated, and
Mhm.
they have. And so, when I first started hearing that claim, as being inquisitive as I am, I would look into them, and I would start investigating those types of claims. And one of the first claims that I saw was the claim that like the CDC had admitted that SARS-CoV-2, the coronavirus that causes COVID-19, um, and let me be very clear about what I mean by that. When I say that the virus causes COVID-19, what I mean is that it's a necessary but insufficient factor in the pathogenesis of COVID-19. So, there's this kind of debate between germ theory and terrain theory, and it's not one or the other, it's both. Obviously, host factors are incredibly important, as we just discussed, and I discussed that with my simplistic definition of health. So, host factors are absolutely important, and to discuss the idea of pathogens like viruses causing illness is not to deny that host factors are determinative. So that's when I say cause, I mean that again. To just quickly summarize that again, it's that SARS-CoV-2 infection or whatever virus infection is a necessary but insufficient factor in the pathogenesis of whatever infectious disease. Um, so, anyways, I started, just looking into these claims. Like one of the first ones that I encountered was this claim that the CDC had admitted that SARS-CoV-2 had never been isolated. Which, you're looking into it, and you can see it's demonstrable that claim was false. And so I just kept investigating these claims, and every time I would look at them, I came away with the conclusion that these claims are demonstrably false, including the claim that no virus, not only SARS-CoV-2. I mean, this was the kind of the first one that I started seeing a lot. Um, Although I had been aware of these claims that no virus had ever been isolated, like measles and so on, but I never really paid them too much attention before the COVID-19 pandemic. I'd never really dug really deeply into those types of claims until 2020. But again, it's just every time I started digging into what these claims were, and then looking at the sources that were being cited to support those claims, every time the claims are actually contradicted by the cited sources. And so, I find it untenable.
Okay. So when you say isolation, um, are you referring to, some sort of purity? Is that what you're getting at with that? Or like, because I guess my understanding of isolation in virology is, isolating the virus out of a host into a cell culture. Is that what you're referring to? Or,
Yeah, cell culture is considered the gold standard, as it's described in the literature of virus isolation. Yeah.
okay.
But cell culture alone is not the only means that scientists have at their disposal to be able to characterize, to identify and characterize viruses. There's also whole genome sequencing is another really important example. So it's not just the cell culture that is evidence of the existence of viruses, but that is what's considered the gold standard for isolation.
Got it. Got it. Okay. And I think that's one of the, I think, where the argument where people really get stuck on the argument there that that's not isolation, right? Is where when we start discussing like Rivers' review of Koch's postulates, right? Like the um, within it says there needs to be some sort of isolation, some sort of purity introduced to a host to prove that that is the causative agent in some sort of infectious disease. I think that's largely where that is applicable to this argument. The other one is that you just brought up is genomics, right? Um, isolation of, and I would love to know if you agree with this. I don't necessarily think that isolation of viral genetic material, especially of a single species of virus, is a possibility.
Well, there's actually this is another another argument that I've seen repeatedly made is that
Sure.
that it's not that you can't do whole genome sequencing of a virus when that genetic material is mixed together with a bunch of genetic material from other things, and that's actually just plain incorrect. They actually have what's called metagenomic sequencing.
Hmm.
And for an example of the utility of metagenomic sequencing is scientists, um, uh, you know, uh, doing this type of sequencing on the gut microbiome to be able to identify hundreds of thousands of different types of viruses that reside in the gut. Mostly, these would be viruses that infect not your human cells, but the bacterial microbiome. So viruses that infect bacteria are what's the word? What are they called? Macrophages. And so, they have done metagenomic sequencing, for that purpose. For example, there's one example that leaps to mind where, that it demonstrates the utility of metagenomics for determining the whole genome sequences of a multitude of viruses, um, without having to first isolate each one individually.
Hmm.
So you can do metagenomic sequencing without actually isolating an individual virus. And this is a big, again, it's just a big misconception, and there's a lot of falsehoods about that. People claiming that that's not possible, but scientists do it.
I think where I would take that is not necessarily that it's not possible to run metagenomic experiments. I think I would question, the validity of the results of the metagenomic process. I think that's where I would like that's where I would get stuck on, because um, Sanger sequencing is the kind of the gold standard of genomic assembly, right? Or genomic sequencing. So, and a purity would be necessary to run Sanger. Like, I don't think you can run Sanger with viruses.
Um, I'm not sure about Sanger sequencing in terms of whether Dr. Singhong Lee would be the expert on that and would be able to answer that question. But
Sure.
in terms of whether they do metagenomic sequencing with that technology, I'm not sure about that, so I can't speak to that exactly. Um, but I think one of the areas of confusion with accepting the results of metagenomic sequencing is just kind of a misunderstanding of how whole genome sequencing works.
Mhm.
Um, and again, I'm not a scientist. I'm not an expert on any of these things. I just know what I know from researching these claims about the non-existence of viruses, and just like digging into those things and spending a really extraordinary amount of time on it. Um, so one of the claims that is kind of made is that, well, one of the claims that I saw early on was that that you can't do whole genome sequencing without a reference genome, which is kind of a logical impossibility. I mean, if you had to have a sequenced genome to be able to sequence subsequent genomes to subsequently sequence other genomes, well, then how did you sequence the reference genome in the first place, right? And so there's something called de novo sequencing, where it's like the very first time, where you sequence something for the first time, and then after that, then you can use that sequence as what's called a reference genome for subsequent, types of genomic sequencing. but there's de novo sequencing, and metagenomic sequencing is a type of de novo sequencing where, again, you don't even need to isolate a specific an individual virus to be able to do that type of sequencing. You can sequence a multitude of viruses or other pathogens directly from a patient sample, for example, using this technology. And so another kind of, I think, misunderstanding is how it works in terms of what it is. And so it's not the case that, as I've seen people claim, that they just kind of randomly assemble the genome from these little snippets of genetic information. It's not randomly assembled any more than you randomly assemble a puzzle when you're putting together a puzzle. I mean, the pieces lock together in one way. This is why, I mean, the puzzle analogy is of course imperfect, but it's useful to kind of simplify the concept of that. The different sequences that they assemble with this technology. There's overlaps in the sequences, and so they can assemble them in the way that they fit together, and so they do fit together in a specific way. And you can't just like take these snippets of genetic sequences and just randomly assemble them to just make up whatever. I mean, it just doesn't work that way. And this is one of the biggest misconceptions that I've seen out there in terms of how it works is the claim that they just kind of randomly assemble this genetic material to come up with whatever they want. And it's just not how the technology operates.
Interesting. Appreciate that. On the topic, the concept of variance comes up in my mind. Because when we're talking about this de novo sequencing, um, I would probably agree that having a reference genome would be helpful in like validating this, of course. That just makes good sense. Um, is it absolutely necessary? You're arguing against that, which I can appreciate as well. Um, when it comes to that de novo assembly, um, we see the variance. That's one of the things, the variations in these genetic sequences, slight variations. I'd love to hear your thoughts on that because to me, in my sort of read of it, is that could be indicative of just an error in the assembly. Like because and maybe you could speak to this as well. I know I'm sorry to throw a few things at you here, but um, in the assembly during that process, I was understood that these platforms, like these softwares that put all of these genetic sequences together that sort of build the puzzle is probability based, um, and that it does synthesize a mass amount of sequences. Like when you run it through these systems, you get sort of a ton of different sequences, and you may get two or three sequences that match, the length that you're sort of searching for. So if it's a certain amount of base pairs, if it's 30,000 base pairs, you have two sequences that were synthesized. So there's two variations of this virus. Um, that was sort of my understanding. Maybe you sort of just did clear that up, but if you want to clear that up anymore, I'd appreciate your thoughts on that.
Well, sure. When they speak of viral variants, they viruses mutate and evolve, right? So the original Wuhan strain of SARS-CoV-2 is different from the subsequent strains that were, or I shouldn't use the word strain because scientists differentiate between strain and variant, but you get my point that the variants that were subsequently circulating displaced the original variant. And so it's as the virus moves through the population, as it infects people within individuals, it can evolve and mutate, and then it spreads to someone else, and there's further evolution and mutation of the virus. And so this is what is meant by variants: is just their mutations of the virus. And if those mutations become significant enough, then they could describe it as as a new strain. But the variants are kind of considered the same strain of virus. It's just that there are genetic mutations there that may be significantly meaningful and may not be. But typically they name the variants of SARS-CoV-2, for example, based on what they consider to be significant um, evolutions in the viral genome. Omicron, for example, having a number of significant mutations in the spike protein that made it that enabled it to escape the antibodies from prior infections and from vaccinations to a significant degree. So that's an example of what they refer to as variants. But I mean, you're right that there can be errors, like in reads and things like this. But there's also you can run sequencing multiple times. Scientists around the world, on a regular basis, you sequence genomes of the virus, and so there's this kind of this redundancy and replication among scientists of the genomic sequences. And so when you there's a very kind of insignificant I would say basis to say that that the outcome of these these computations, which, because of course whole genome sequencing involves computation, because as you explained, I mean, it's something that the human brain can't do. It requires a computer. So they have software for that, and so it's not the case that you're just getting these errors and it's just spitting out whatever. Yes, it's true that you can have read errors and things like that, or assembly errors. But at the same time, when scientists are replicating the process over and over and over again on a daily basis, then there's kind of a limited room. There's kind of limited room to say that, well, we don't accept the outcome of the genomic sequencing, right? As many people do, and they did just dismiss whole genome sequencing altogether. And I just find that just untenable and unsustainable.
Okay. It seems as though this conversation is quite far away from life. We're in the computer with this. Um, there may be, this is the interesting thing. You know, if we accept that the genomics, the metagenomics, is an accurate representation of what is in some sort of sample biologically, um, it still seems as though it's quite far away to make claims um, about these little particles. That where the isolation process is not really some purity, especially when it comes to making claims about their pathogenicity. You know what I mean? Like if it's going to cause disease, if it plays a role in disease. Um, I'm curious what your thoughts are on that, because it just, to me, it seems like it's quite far away from life. We're not even just in the test tube now. We're in the computer, and we're going off the assumption that this computer represents life, like these models represent life. And I guess there's a long history there, but just
Sure.
curious
Well,
to hear your thoughts there.
yeah, the question of whether a virus causes disease or is pathogenic is a separate question from whether the virus exists. So you have to separate
Certainly,
these two questions.
certainly.
You can prove that a virus exists. It doesn't mean that it causes the disease, that people suspect it causes. And this was acknowledged in the initial papers on SARS-CoV-2, where when they first isolated and sequenced SARS-CoV-2, those initial papers, expressed their opinions that this was the likely culprit. This was likely what was causing people's illnesses, in Wuhan, China. But they said right in there that Koch's postulates haven't been fulfilled yet. They acknowledged that it was yet to be determined. So they had found a key suspect, essentially. They found a SARS-like coronavirus, and so that this was, in their opinion, probably not a coincidence. That this was the likely cause of this illness that they were seeing. which was a reasonable conclusion to draw. But Koch's postulates were subsequently fulfilled. There are animal challenge studies, hamsters, um, uh, macaques, different species of animals that they did challenge studies and demonstrated the transmissibility and the pathogenicity of SARS-CoV-2 in those studies. In fact, there's even been a human challenge study. So, the first question is, what is the cause of, when with this is what investigators were asking themselves. There's this strange pneumonia-like illness. What's causing it? They took patient samples. Typically what they'll do is, you take the patient sample, you filtrate it or centrifuge it to purify it. There is a purification step. And this is another false claim that's widespread out there, is that there's no purification. Well, but there is. They purify the sample. And then they take the supernatant from that purification, and then that's what they inoculate the cell culture with. And then there's another false claim out there that they never do controls. Scientists never use controls when they do cell culture. Also completely false. They do use controls, and so they have the inoculated cell culture, and then a control culture that's given the exact same treatment, only without the inoculation with the supernatant from the patient sample. They treat the cells. They have to give food for the cells. There's antibiotics to prevent bacterial contamination in the cell culture. So they're given the exact same treatment, except they're not exposed to the supernatant containing, potentially containing a virus. And then they watch and they observe the cell cultures side by side, and to observe for cytopathic effects, which would be indicative of the presence of a virus in the infected sample, in the affected cell culture, compared to the uninfected control. And so they can compare, you have this cell culture here that's got cytopathic effects, and the uninfected control does not. Well, that's an indication that there was a virus in the patient sample. And then they do further analyses from there to characterize the virus, including you can look at it underneath an electron microscope. You can do whole genome sequencing, et cetera.
Can I chime in? Because we'll
Yeah,
get to the
absolutely.
electron microscope. I just want to chime in on that. I'm curious about your thoughts on like Stefan Lanka's experiments, in which, in his experiments, claim that he showed that the procedure of using um, of starving the cells, as well as using antibiotics, or streptomycin, I think is commonly used, actually was able to create cytopathic effects. I'm curious about your thoughts on those experiments, if
Sure.
you've
My
looked
first
into them
thought
at
is
all.
he should publish his research.
Sure.
My second thought is he's not explaining why scientists all around the world have done this experiment over and over and over again. And guess what? They do use controls, and they can compare uninfected controls that don't have cytopathic effects. And he's claiming, well, no matter what, you're going to get cytopathic effects just from how they treat the cell cultures. It's just
Mhm.
demonstrably false.
I think one of the big problems, from my read of the literature at this point, a lot of the studies on cytopathic effects, when it comes to describing their use of mock infection, um, which is what you're talking about here as the control. Mock infection would be like similar or same setup in theory. It should be the exact same setup, same parameters, same cell culture, same whatever, without virus or sample from the individual added. Um, one of the problems is that they say, oh, mock infection was used as a control, and they don't actually describe what was done in the mock infection. Now, maybe they think, maybe the scientists at that point think, we shouldn't have to, if you're reading a paper, you should know what a mock infection is. We did the exact same thing. Um, Personally, when it comes to the literature, I think that is something that should be done. I agree with Stefan Lanka's work, like you mentioned. I think you should publish this research. I agree with that. I believe in transparency, and one of the things I learned about the use of the scientific method in writing scientific papers is that the method section should be extremely, extremely clear. that you can reproduce the results, because reproducibility, of course, is the cornerstone of science, as you've alluded to a couple times here.
Mm.
That is one of my main problems, is that
Yep.
they're not describing the steps in mock infection, and I think that's where a lot of people get hung up. That's a place that I get hung up on, definitely.
Yeah, there's this claim out there that when they refer to mock infected cells, that's not a control. But again, it's just simply false. Mock infected is literally defined as an uninfected control. So it's given the same treatment. The only difference between the infected and uninfected cell cultures is that the uninfected control is not inoculated with the supernatant from the purified patient sample. It's that simple. That's what mock infected literally means. And so, when scientists use the term mock infected, yeah, of course they don't feel the need to define their terms because it's just understood within the scientific community. Everyone knows what a mock infected culture is.
Mhm.
It's the literal definition. So this is, again, it's just kind of it's one of those claims that are made out there that mock infected is not a control, but that's literally the definition of mock infected culture. It's a control.
Gotcha. Okay. Yeah. Um, what was I gonna ask there? So I'm curious. You know, there's a group out there now that it's coming to light, and it's not published yet, but I hope to stay in contact with you, and would love to hear your thoughts on this. They've reproduced Stefan Lanka's experiments, um, and I think they're going to publish their research, and when it is more out in the open, I'd love to hear your thoughts on it. Is that something that is convincing to you at all? Like, if there is some sort of step in that process that does produce, through electron microscopy, visible, viral-like particles or exosome, whatever you want to call them, extracellular vesicles visible under or plaque forming units and cytopathic effects. Is that something that's convincing to you?
Um, they would have to explain why there's so many published studies of cell culture experiments in which the uninfected controls didn't have cytopathic effects. I mean, this is their claim that, oh, well, the cytopathic effects, and this ties right into their false claim that scientists don't use controls. So it's I'm sorry, but it's a hoax. It's a hoax claim. Because again, they're not explaining why scientists all around the globe do these experiments, and they do use controls. And guess what? They can compare the infected and uninfected controls, the infected cell culture with the uninfected control culture, and there's no cytopathic effects in the uninfected control. And so it's not the case that the treatment given to the uninfected control is what's causing the cytopathic effects. It's observably not true. So they can, they could publish this supposed experiment of theirs. I'd love to see it. If they want to publish it, I'll take a look. I'll look at their methodology. First of all, are they doing the same? Are they following the same steps that scientists all around the world are doing? That's number one. I want to see it. Show me. And then they would have to explain why they're getting a different result from everyone else all around the world.
Mm-hmm.
So it's just my view of it is that it, just like other claims, just like the claim that the CDC admitted SARS-CoV-2 wasn't isolated, just like the claim that scientists never purify patient samples before they do cell cultures, just like the claim that they never use controls when they do cell culture. These are all hoax claims. They're false. And these are all the types of claims that are used to support this belief in the non-existence of viruses. And so my point of view is this: it's that, if somebody wants to propose some alternative hypothesis for the pathogenesis of what are called infectious diseases, I'm all ears. But it's a situation where, you have to accept the hypothesis that best fits the totality of available evidence. And the entire body of relevant scientific literature is consistent with this idea that viruses exist, and some, not all, some can cause diseases in humans. And I'm not aware of a single study, not a single one, including all of the studies that are cited by proponents of the no virus theory, the no virus belief, including those papers. There's not a single paper that I've ever encountered that supports this idea. Not one.
Hmm.
Not one.
Hmm.
All of the medical literature supports the hypothesis that viruses exist and they can cause disease in humans. So it's a matter of competing hypotheses. And which are you going to accept the hypothesis that has no evidence, or the hypothesis for which literally all of the evidence points to? And so I just have to accept the hypothesis that literally all of the scientific evidence points to.
Understood. Yeah. So electron microscopy is another method that's used, of course, to visualize viruses.
Yep.
Um, Maybe you could speak to that a little bit, and then we can dig into that.
Sure. Well, there's that claim too, as well, that what scientists, because there's published papers, and you've seen it in the media where they show electron microscopy images of the SARS-CoV-2 virus, for example, with its characteristic spike proteins, which give it this why it's called coronavirus. Corona meaning crown in Spanish, or Latin, and so it's got that. It's called a coronavirus literally because of the appearance that the spike proteins give to it, of the appearance of a crown. So this crown-like appearance is why it's literally called a coronavirus. And so we've all seen those images, I think. And there's a claim that, well, that's not really a virus that scientists are photographing. It's exosomes. Well, this gets into additional claims that are demonstrably false. So, for example, the claim that, well, scientists can't distinguish between exosomes and viruses. And you can go and look up that paper that's cited to support that claim, and you can see that the entire thrust of that paper was that actually it's true that if you just are using electron microscopy, they're difficult to distinguish. Granted. But the whole thrust of the paper is how scientists can and do and should distinguish between exosomes and viruses. So there's additional, analyses that scientists do to be able to distinguish. Yes, just looking at the electron microscope images, an exosome could look very similar to a virus. Absolutely. And so this is the point that was made in the paper, but it's completely that, statements like that from the paper are taken completely out of their context. And if you just go and read the whole paper, the whole point of the paper is about how scientists can and should differentiate between the two. So the claim again, the claim being made to the claim that this paper is being cited to support is that scientists can't distinguish between the two. And you go and you read the paper, and it talks all about how scientists can distinguish between the two and how they should do that. So this is an example of kind of like these hoax claims that are out there. And again, claims that are actually contradicted by the sources that are cited to support them. Yeah.
Gotcha. Yeah. Nah, and I appreciate that honest take. I would agree that with what you're saying there about electron microscopes. It's not enough. It's a piece of the puzzle
Precisely.
for virology's
Yeah.
claims.
Yeah. Yeah, again, that's a really important point. It's not just about isolation, or just about electron microscopy, or just about whole genome sequencing. There's a variety of different tools and analyses that scientists do to be able to identify and characterize viruses.
Mm-hmm. Yeah. Um, Definitely. And the last big one is immunology, serology, antibody research. That's sort of the last big one used to identify antigens, things like SDS page, electrophoresis, stuff different. There are many different types of ways to do that. Western blot is a big one. There's a ton of different types of blots too. Anyways, that's another big one, and that's one of the things. It's all very reliant on each other. to sort of prove each other correct. That's like you were pointing out there. Um, with antibodies, one of the big things, and this may be out of our scope here, um, one of the main claims. Curious if you've heard about this, have any thoughts? Um, antibodies displaying all antibodies displaying some sort of non-specific behavior, and so shouldn't be used again to as a you know gold standard to detect whether or not there is some sort of virus or viral material present. I'm not sure if you've encountered that claim whatsoever, but
So non-specific in terms of that an antibody might bind with. So in other words, the assumption might be, okay, well, this antibody bound with this particle, therefore that particle is such and such virus, when actually that antibody might also bind with other particles. Is that what you mean? Is that the
yeah,
thrust of it? Okay. Actually, I don't know that I've heard that. But that's a legitimate uh, point that, yeah, sure, not antibodies might be not necessarily specific. There might be non-specific, but in general, um, antibodies do have specificity to whatever they are developed in response to. So, for example, when you first experience an infection with SARS-CoV-2, um, and I'm just picking that because it's kind of at the forefront of my mind when I talk about this
sorry.
issue. But pick your virus. But I'll speak on that also because I've researched it so extensively.
Sure.
Because one of the reasons I spent a lot of time researching the immunology on SARS-CoV-2 was because of the claims that were, throughout all of 2020, in the summer of 2020 through the fall, right up into the point when the vaccines received emergency use authorization. We were bombarded with all these headlines claiming that people were losing their immunity to COVID-19. And I was writing to debunk all those propaganda claims that were, of course, designed to create fear and to manufacture consent for the experimental vaccines that were under production. But anyhow, so I'm just picking this particular virus because
Okay.
it's in front of my mind when we talk about this issue, and also because I know so much about the immunology of it. But pick your virus. But I'm just going to pick that one. So when you're infected for the first time with SARS-CoV-2, it although there is some level of cross reaction with prior infections from common cold coronaviruses. So your body does have some, because you've experienced coronavirus infection with common human coronaviruses that cause the common cold. You've got some immunity to those viruses, and there is some like cross reaction in terms of your immune response from that past experience. Not necessarily terribly protective against SARS-CoV-2 because it is quite different from the common cold coronaviruses. And so it can take a couple weeks for your body to actually develop antibodies to that infection. It can take several weeks. But the body starts going through this process of producing antibodies. And, it might start out producing antibodies that really don't have high affinity to the virus. But in time, the antibodies that are effective in binding to and neutralizing the virus are selected for. And so the body learns and trains itself, the immune system trains itself in how to produce an effective response. And so, and then what happens is, once you have essentially cleared the infection with SARS-CoV-2, also what happens is you develop, which is typical for infectious diseases, is you develop a long-term immune memory. So you have long-term bone marrow plasma cells, and this is one of the claims that they were making in the media and the headlines where people were losing their immunity, and that was based on this observation that, well, people had high level of antibodies after the acute phase of infection, and then it just started to plummet, and so people were losing their antibodies. And so therefore they were losing their immunity. And this was a bunch of ridiculous nonsense. It was like contrary to like immunology 101. And again, I'm no expert, but it's just like basic immunology that, well, that's completely normal for that to happen after the acute phase of an infection. It happens with every infection. Where, yeah, you don't maintain that high level of antibodies after the acute. Yes, there's always going to be a decrease in the antibody levels, but every study that looked at it after that found a continuation of detectable antibodies in people's blood who had recovered from COVID-19. And scientists also confirmed that, yes, that infection induces long-term bone marrow plasma cells. So you have long-term immunological memory, so that if you ever encounter that virus again, your body, your immune system doesn't need to learn all over again how to fight it off. It already knows. And so those cells are able to rapidly produce antibodies again that are high affinity to that infecting pathogen. And this is where, we talked about variants earlier. This is where, another issue with the variants is that, so people's immune memory would start producing antibodies that are specific to whatever variant that they were infected with. Let's say they were infected with the original Wuhan strain, and later they were infected with Omicron. So their body was able to rapidly produce antibodies, but they weren't as specific to the Omicron variant because they were developed in response to the original Wuhan variant, right? And so, but here's another really important thing is that with natural immunity, the body is able to update itself. And so people who are, maybe had a reinfection with Omicron, their immune systems were able to rapidly update that antibody response to be specific, to have greater specificity for the spike protein of the Omicron variant. And this is something that people who received the vaccine weren't able to do, because with the vaccine, there's a problem that's known as original antigenic sin, where the initial priming of the immune system can prejudice the immune response to any subsequent exposure to whatever pathogen. And so people whose immune system were primed by infection have a robust, long-term adaptive immune response, whereas people whose immune systems were primed by vaccination don't. We've seen, of course, there's the rapidly waning immunity with the COVID-19 vaccines, and then there's this problem of original antigenic sin, where they notice that if they did experience an infection, what they call a breakthrough infection with, say, the Omicron variant, that the antibodies generated don't adapt. They stay fixated on producing an immune response to the extinct spike protein of the extinct Wuhan strain, at the opportunity cost of not producing a more specific response to the newly infecting variant. And so that's known in the literature as original antigenic sin, or sometimes it's called immune priming, which I think is a really stupid name for it, because every immune reaction to any new pathogen is immune priming. So it matters what is the priming? What is is it priming from infection, so natural immunity, or is it priming from vaccination, which is different? And so just immune priming is kind of a very nondescript label for this phenomenon, but it's also called original antigenic sin, which I think is more fitting and a better description for it.
Cool.
But anyhow, yeah, I think so this kind of speaks to the we were talking about variants earlier, so that kind of ties together in terms of antibody responses, and there is a question of specificity of antibodies to a pathogen. But the whole aim in terms of the immune system's function is to produce antibodies that are specific, and of course they're not always, and this can be a problem. And it's why people can get reinfected with variants, even though they've had an infection before.
Non-specificity of antibodies doesn't necessarily contradict that either, though, because if we have they talk about the big five, that's what we learned about in my introductory microbiology classes. It doesn't necessarily go against the theory at large of, you know, having some sort of immune response either, which I think is interesting. Um, Used as a tool, it is a source of irreproducibility in the literature, which I think is problematic. And there are a lot of of companies out there that are trying to remediate that. Like it's a huge problem in science is the non-specificity um, of antibodies because it's used across the board, in science and scientific research. So um, something to look into. I'm really curious
Are you?
about.
I don't know. I don't know what you're thinking of, but I'm thinking of, like, for example, um, maybe this is completely different from what you're thinking, but like, for example, they're trying to develop vaccines for Lyme disease, and the FDA is using antibody tests to kind of determine, whether those vaccines are effective. But the problem is that, um, a lot of people who are infected with Lyme don't develop antibodies, and so if you're just looking for
Mhm,
antibodies to Lyme and it's negative, it doesn't mean you don't have Lyme disease, for example.
mhm,
If you look at vaccine licensure, there's a problem with looking with the use of antibodies, because the whole purpose of the vaccine is basically to cause the body to generate antibodies. But of course, antibodies are not the immune system's only response to pathogens. There's also cellular immune
sure.
responses and T cells. And SARS-CoV-2 is actually a very good example of how the cellular immune response is actually more important than the antibody response. And this
Hmm.
is something that the public health establishment only seemed to discover after they had to finally admit, after it was just absolutely clear in the data that the vaccine effectiveness against infection waned rapidly. And suddenly they discovered cellular immunity. And they were like, oh, but don't worry, you might get an infection, but you still have protection against the disease. And your symptoms won't be as bad because of you have T cells. And suddenly they discovered T cells, right?
Mm.
But of course, this is something that they had always known about, and they just never educated the public that it's not about antibodies. It's not all about antibodies are important, in terms of preventing a circulating virus from being able to bind with cell receptors and enter and penetrate the cell membrane to gain access into the cell, and then using the cell's mechanisms to replicate itself. Because
Yeah.
the virus is an intracellular parasite; it requires host cells to replicate. And again, that's why scientists use cell cultures to isolate viruses. But antibodies are just one factor in a broad range of immune responses that are important. So I'm not sure if that's along the lines of what you were thinking, but we might have some agreement in terms
Definitely.
of, if you're just looking at antibodies, it can be very deceptive. Or when they say that, well, if you have a certain level of antibodies, that means you're protected. Well, that's not necessarily true. You
Yeah.
can, in fact, with SARS-CoV-2, study after study was showing that that patients who had severe disease was correlated with a high level of antibodies, whereas patients who had very mild symptoms or no symptoms at all, they had an infection but no disease, and that's another important distinction. You can have an infection without
Mhm.
disease, and so they were finding that patients who had who their immune systems just absolutely handled the virus without hardly any problems whatsoever had very low levels of antibodies, and this is explained by the importance of the cellular immune responses. And so there was actually the contrary correlate. They want us to think of you need a high level of antibodies to be protected against this disease. In fact, in terms of natural infection, the opposite was actually the correlation observed, were that people who had high levels of antibodies were deficient in cellular immunity, and so they developed more and more antibodies. But those were also the patients that had the most severe disease. And
Very
so,
interesting.
absolutely, if you're just looking at antibodies in this context, there's a lot of misunderstanding and misconceptions about that. But I don't know if that has anything to do with the line of
Well,
thought
that's an
you were on.
interesting observation at the very least. I think that's something that merits a little more digging into for sure. That's because that's a really interesting observation to say the least for sure. And it sort of circles into one of the last things I want to ask you about. I got two more things if we got time, but um, the one thing I want to ask you about is the PCR test. Um,
Yeah,
you know,
glad you brought it up.
yeah, I think that's an important part of our discussion for sure. So maybe I'll let you have the floor and then I can chime in.
Well, sure. I mean, as soon as you mention that, again, my thought is there's a lot of confusion out there. I've had so many of my own readers. It's not so much anymore, but in 2020 and 2021, throughout that period of time, just so many of my readers were so confused about kind of the distinction, because they were seeing how the PCR tests, the the tests that were being used to diagnose COVID-19, there was systematic fraud in the way they were utilized to diagnose COVID-19, because the PCR, those types of PCR tests, just detect viral fragments. So they can detect RNA from the virus, but they don't look at the whole genome sequence. So they can detect just fragments of viral RNA. And you can have fragments of viral RNA in your body, and it doesn't mean you have an infection. It could very well mean that maybe you were infected before, and your immune system has already fought off the virus, and you don't have an infection at all, and you're completely symptomless. There's no infection. You're not contagious. You're not a threat to anyone. And they were calling people, who had just like fragments of RNA in their system. They were calling them COVID-19 patients. And well, that's false. That and to utilize these tests in that way was really systematic scientific fraud in the way they and the other thing is you could even have an infection, but that's not necessarily the cause of the disease.
Sure.
Which I'm sure you agree with, that somebody might have the PCR test might be positive for viral RNA from SARS-CoV-2. It doesn't mean that that's the cause of the disease. Again, there might not even be an active infection in that case.
Mm-hmm.
And so to describe anyone with a positive PCR test as a COVID-19 case also overlooks the distinction between infection and disease, because again, you can have an infection and not have the disease. The disease is the symptoms. That's what a disease is. The clinical disease is the syndrome, the symptoms that you get from having an infection. If you don't have any symptoms, you don't have the disease. So the disease doesn't actually develop, and that's another important distinction. So the way that they were counting COVID-19 cases was really premised on this scientifically fraudulent use of these PCR tests. And so a lot of my readers and a lot of people that I saw, it's on social media and whatever, engaged with, were just like, they were seeing that, and then they somehow like took that over to the use of genetic sequencing, genome sequencing, which uses PCR technology. And so they were kind of saying, well, so the PCR tests are being used in a fraudulent manner, and therefore, the whole genome sequencing uses PCR technology, that must be fraudulent too. And so people were kind of making this leap of logic in because they were hearing that type of thing from others who were kind of propagating these types of claims. And so I kept finding myself having to kind of clarify, hey, look, yes, I'm saying that whole genome sequencing proves that this virus exists. It doesn't mean I'm saying that these PCR tests are being used in a legitimate fashion to diagnose COVID-19. It's like people would make that assumption and like attribute that kind of argument to me. And I'm like, no, that's not what I'm saying at all. So anyway, when you bring it up, and I say I'm glad you brought it up, that this is kind of what's going through my mind because of that experience of mine, dealing with lots of people on social media, or even my own readers emailing me and kind of either just confused about it and asking me, or like challenging me and confronting me about that. And so it's really important to understand the difference between like those PCR assays that were used for improperly for diagnosis, and just the PCR technology itself, which is a legitimate technology. And so people need to understand that distinction.
And I'm glad we agree on that, and I think I'm curious if you looked into this at all, but they were running the tests at a much higher cycle threshold than is scientifically acceptable.
Well, that's the other factor in that, is that they were running the PCR test at such a high cycle threshold. So for your viewers who aren't aware, so the way the PCR tests work is actually, so it's reverse transcription polymerase chain reaction, so RT-PCR. And so they because the virus is an RNA virus, and so they have to reverse transcribe the RNA into DNA, and then they use cyclical amplification of the DNA up to a what they would consider a threshold of positivity. But they were running the test at such high cycle thresholds that they were turning up positive for just tiny fragments, like tiny amounts of viral RNA that did not represent whole viable virus. And it's such high cycle thresholds that it was known that that didn't represent that.
Yeah,
In all probability, they were running the test at such high cycle thresholds that they knew that that it probably did not represent somebody who had an infection and somebody who was contagious, and they knew it.
absolutely.
So, and
Absolutely.
this again, I'm glad you brought up the cycle thresholds, because again, it just speaks to how widespread this scientific fraud was in terms of diagnosing what they called COVID-19 cases. And then, of course, anyone who was a COVID-19 case who died, they considered a COVID-19 death.
Yeah, of
So
course.
the amount of scientific fraud in that regard was, absolute. But again, a lot of people were confused about that when it came to like
Yeah.
the PCR technology being used with whole genome sequencing, and
Mhm.
just kind of not understanding the difference. Um, but yeah, I mean,
It's
I'm
a different
absolutely in
discussion.
agreement about the
Absolutely.
systematic fraud involved with PCR tests.
Yeah. Okay, that's great. The last, if I can ask you about one more thing, I'm curious about your thoughts on like real world contagion studies. And the big one for me, and the first one that I ever read that was the most convincing for me on sort of the train versus germ argument was the Gallups Island study that was done back in by Rosno back in 1919 for the Spanish flu. And he took a bunch of, I'm not sure if you're familiar with the
My
study, but
recollection was 1918, but I think pretty sure we're talking about the same study. Yeah.
I think we're talking about the same study. 1918 is correct. Yes, you're right. But yeah, I'm curious. So I'd love to hear your thoughts on that. The listeners are quite familiar. It's the controlled contagion studies, essentially, of
Yeah, that's the
trying to
one
prove some sort of contagion.
I've most often seen cited. Um, but I don't recall. It's been some times. I think I've looked at that whole study. Pretty sure I've managed to get a hold of it and look at it, but I don't specifically recall. Um, Other than that, the important point to make is that in 1918 they knew practically nothing about viruses, and they knew practically nothing about viral transmission and contagion, and they had some sense that there were these tiny particles that were, transmissible and could infect people. But their knowledge of virology and immunology was just primitive and basic. And we've had, over a century of scientific research since then, with many, many studies demonstrating transmissibility demonstrating existence of, as well as contagiousness of viruses, including, as I mentioned earlier, with SARS-CoV-2, there's that claim that Koch's postulates, as understood today. Of course, Koch's original postulates were just plain wrong. So, and then you mentioned Rivers' kind of, modification of Koch's postulates, and Koch's postulates have been kind of modified over time, to become, more specific and more accurate and more valid in terms of how do you determine whether, particular agent causes a disease. So when I refer to Koch's postulates being fulfilled in terms of SARS-CoV-2, I'm talking about, as scientists understand Koch's postulates today, and the modern research methods and modern understanding of the steps involved to actually demonstrate causality. So, there were, animal challenge studies. And even as I mentioned, there was actually one human challenge study with SARS-CoV-2 where they deliberately infected human subjects, and then observed the pathogenesis of disease in those patients. Um, so yeah, I mean, this claim that people, I see people citing that 1918 study as though that study proves that there's no that the flu wasn't contagious, but it doesn't prove that. Um, I mean,
Gotcha.
I
Can
guess
I tell you something
that's
about?
my main point is that study doesn't prove that.
Sure, got it. And that's good. And I want to tell you something about my experience in university. because I just graduated from my degree 2022. So when I was in microbiology, when I took all my microbiology classes, they taught Koch's postulates. And I could appreciate what you're saying. Like I can. You know, that's an area of distrust for me personally.
Okay.
And I think that there's this it's not right in my mind that university students are going in, they're learning about Koch's postulates as being the gold standard way that things are approached, when now in scientific fields something different is being done. I think that's a
You
huge
mean they
problem.
taught you like Koch's original postulates?
Original postulates,
As
the
though that's
core
still
postulates,
valid
is
today?
what we were taught in multiple classes, not
Wow.
just one micro and
Okay,
high
okay,
level up
so
class, 3,000, 4,000 level courses. They were teaching Koch's postulates.
okay,
And
so just as an example of what I mean when like that his original postulates are invalid and now known to be incorrect is, for example, he had made an assumption that if there's an infection, therefore there will be a with a pathogen, therefore there will be a disease, and so um,
we know that's not
you will not
true.
find, in other words, you will not find that pathogen in anyone who doesn't present with the disease,
Yeah.
which is completely false and was subsequently disproven, which is why one of the reasons I think Rivers was correcting the original Koch's postulates and updated them
Yeah.
with the improved knowledge of scientific thinking. So I'm really shocked to hear that they were actually teaching the original Koch's postulate
Not a
as
word
though
of
they hadn't
Rivers.
been like refined over the year, over a century of scientific thought and knowledge, that hadn't been refined. That would also, if I was you going through that experience, would also create distrust with me, because that's crazy. Yeah, that's
Hmm.
crazy. But I guess
Yeah.
it shouldn't surprise me that they would teach something like that in school.
So the other amazing thing was, like, I was taking genetics too. My degree was largely genetics. I took biochemistry and molecular biology, so it
Okay.
was mostly genetics. You know, I was in a genetics course during the real height of COVID, and we were learning about the intricacies of the polymerase chain reaction test. And my professor probably said 33 cycles 1,000 times. We were tested on it. It was in our exams, midterms, everything. 33 cycles, 33 cycles. When I brought out the CDC document that said that labs should run at 40 cycles, she almost kicked me out of the class. She lit me up in front of the whole class. And anyways, my experiences certainly, you know,
Well,
influenced my view in life a lot.
Fauci himself
But
admitted that when you run, even at 33, forget 40, even at 33 or 32, that's probably a non-viable fragment of RNA.
yeah,
So
yeah, so I think, and I really appreciate you for what you do, because you're going firsthand, you're learning about this, you're looking at this stuff. I know we disagree on a few points, but I think generally we agree on quite a bit as well. We agree.
yeah,
We
I think it's important to focus on those, the common ground and the areas of agreement, and at least to establish a basis for discussion,
Mhm.
right? So I appreciate your
Absolutely.
approach and inviting me on respectfully and with an open mind to have this discussion.
Yeah.
And, I think this is actually a great example of the type of discussion that can be had on this contentious issue. I've had a lot of bad experiences, to be honest with you, dealing with this issue with people. I've had probably more hate mail about that issue than I have on, because before I was doing stuff on like health freedom, I was, writing pretty much entirely on like foreign policy matters, and I think I had more hate mail for challenging these claims about the non-existence of viruses than I did for like writing about the Israel-Palestine conflict. So
Yes. Imagine, eh?
it was quite disturbing to me, to be honest. So I appreciate this conversation and the respectfulness of it, and I'm glad that we're able to find so many points of agreement. Yeah,
No, and I completely agree with that point too. Um, with what you're saying, there is a level of hostility that I'm completely against. And I think it's just a problem in the health freedom movement. Like it's hindering progress more so than anything. We talk about how the establishment suppresses us all, whatever. Not being able to converse within the sort of health freedom community is a bigger problem, certainly indicative of a much larger issue at hand. So,
yeah.
um, I'd love to do this again sometime. I'm sure we'll be able to. Maybe we can next time we can delve into some studies together. I think that could be quite interesting to do as well. Like if we get into the more nitty gritty stuff, if you're open to another conversation in the
Sure.
future.
Well, with the understanding that you've got more educational background in those areas than I do, because I had a communications degree. I went to school for film and video, is what was my emphasis. So I didn't have I had some biology and I had some. I don't
Yeah.
think I had any. I had one class on. We discussed genetics in my anthropology class, as I recall, but I never studied any of these fields. Again, I'm not a scientist, so I don't have that background. But if you want to look at some studies, I'm happy to give my layman's perspective on them.
Well, I think it would give an interesting discussion, nevertheless. And to be quite honest, I think you're brilliant too. And you've done a lot of work, so I really appreciate that, honestly. And I appreciate you coming on. Thank you so much for your time today.
Sure. Appreciate you having me.
Um, and I just want to hear maybe where the listener can learn more from you. Like I've read your blog, very informative, a lot of great information there. You could highlight that anywhere else that they can find you on the internet and how they can support your work too.
Sure. JeremyRHammond.com is my website. It's where I publish almost all my content. And when you visit JeremyRHammond.com, just sign up for my email newsletters. And of course, I'm on social media. But you can get the links to all my profiles and from my newsletters. So yeah, just go to my website and sign up for my newsletters to stay updated with my work. I cover foreign policy issues, matters of health freedom, speaking out against public vaccine policy and the systematic violation of the fundamental human right to informed consent. Of course, I was against the lockdowns from the beginning, speaking out and exposing the lies that were being told to manufacture consent for the lockdown madness and their coerced mass vaccination endgame. So I write about quite a wide variety of topics, also economic matters and economic freedom, with particular emphasis on the role of the Federal Reserve. So I've written on a wide variety of topics over the years. You can find all my stuff at jeremyrhammond.com.
Awesome. Yeah, we'll put all those links down below for the listeners. Again, appreciate your time. Thanks so much.
Thank you.
I want to thank you all for listening. You should all know that this is not medical advice. This is for your informational purposes only. But also remember, we're all responsible, sovereign beings capable of thinking, criticizing, and understanding absolutely anything. We, the people in the greater forces, are together self-healers, self-governable, self-teachers, so much more. Please reach out if you have any questions, criticism, comments, concerns. Can't wait to hear what you all have to say about this episode. I think we'll have some lively discussions in the Instagram DMs after this one for sure. And I know, Jeremy's open to conversing as well. I would certainly ask that everyone remains respectful. I'd hope that that's something I foster in my community. So, I know he's open to chatting about this stuff. So, you know where to find me on Instagram if you want to reach out. You know where to find him now. And listen, I really appreciate you all for listening. If you enjoyed, give us a like, share, comment, subscribe, review, whatever your platform is. You know how to help support us. So that goes a long way. I really appreciate it. Sharing, especially. And just remember, there's two types of people in the world: those who believe they can, those who believe they can't, and they are both correct. Remember that. Thanks, guys. Take care.